Welcome for today's presentation in the Access Mode exhibition. As part of the public program, today we have a debate where I am honored to be the moderator between scientists and artists. So let me introduce first our guest, Ekaterina Skorb. She did her PhD in physical chemistry, and now she is full-time professor in the Infochemistry Scientific Center of ITMO. And she also made a postdoc in the Alexander von Humboldt Fellow at the Max Planck Institute of Colloids and Interfaces. And well, I also have the honor to call her my scientific supervisor for the exhibition of Access Mode. And thank you, Katerina, for your time.
Thank you for great work.
Thank you. And then also, I'm very honored and pleased to introduce Lyndsey Walsh, American artist, designer, researcher, and also writer of art and science based now in Berlin. And she made her bachelor in individualized studies from the Gallatin School of Individualized Studies in New York. And she also was awarded with the Master of Biological Arts in Symbiotica in the Western Australia University. And now she's also part of the ITMO family, giving workshops and a full course in ITMO and the rest as part of the residency in the gallery air. So welcome, Lindsay.
Thank you. Great to be here.
Well, the main topic of our discussion is the artist and scientist collaborations. The topic of art and science is sometimes still very polemic, and especially we cannot forget that we are humans and we have very intricate and very complex relationships. So that's what is important to understand, how to make successful collaborations between artists and scientists, to understand the difference and the similarities in the approach for research. So I want to invite Ekaterina to share with us a little bit of her research in the lab, especially to motivate and inspire maybe the artists that will be watching this to imagine and fantasize what kind of artworks we could do together. So Ekaterina, the virtual stage is yours.
Thank you, Laura. I'm starting to demonstrate the screen. Hope it works. I try to do without videos, which is a pity, of course, but it should work. It should work at least with a demonstration. And in general, Laura have already mentioned that I spent a major of my scientific life in Germany, in Max Planck Institute of Colloids and Interfaces, and I was a visited scholar at Harvard with George Whitesides. And to be successful in science, we touch in interdisciplinary areas, and this idea of being interdisciplinary community, I agree for my position in ITMO University for starting with computer technology and control, internet technology, new area of chemistry, and in particular Infochemistry, which is a fundamental experimental area of chemistry which deals with information storage and processing on a molecular level. And recently we also discussed a lot robotization and digitalization of chemical technology. And here, of course, surrounded should be, if I can do chemistry myself, I touch a lot of physics. My defense was in a physical chemistry. We published a lot with biological application, but to go to understanding with IT and mathematics, I moved to the best, I would say, IT university all over the world, and people know that Russia is famous, like our students winning Google Olympiads or Olympiads in the direction of mathematics. And idea was that we can suggest that chemistry can also be used to solve a difficult question of making synthetic cell chemical computer when we collaborate a lot with mathematicians and find out the mathematical dependence. And now we are in a city center of St. Petersburg, and we find out that to have this, we also need to develop not just science, but also education, because young people much easier take this idea of interdisciplinary and try to touch various topics, and that's why we also develop a specific master program. It's of course in English, because science is in English, and we are open to welcome people all over the world, and we discuss these complicated areas, but it's science together with education, and there are two basic specialization of digitalization of chemical technology, and in general fundamental one, basics of molecular organized system and infochemistry. And ambitious aim, we are talking about of our science, everyone, the best scientists, the leaders of Russia and the world, since the topic is so ambitious to code information with molecules, to change idea in IT world, the people like, for example, Jean-Marie Len wrote us a letter that he happy to be an advisory board of our center. Robotization of the chemical technology give us possibility to start a co-laboratory together with Daria Andreeva-Boyner and Professor Konstantin Novoselov, Nobel laureate in physics, and we signed with them a joint laboratory. We have one more laboratory with Liverpool University, organized meetings and of course applied the project. But in general, in a center, there are several scientific groups: Infochemistry, chemometrics, computer chemistry, computer digitalization of food, bioelementology, biomimetic materials, and of course group of IT and engineering, as well as joint laboratories. And idea is that we are taking our chemistry, apply series of complexity and networks, model approaches for IT and mathematics with proper algorithm, and we go to other understanding of the reaction diffusion system and how we separate our scientific aim. Usually we have a complex problem which is significant and important. In science, we have some approach. An approach which we are talking about in majority is controlling both reaction and diffusion, and we discuss a non-linear chemical system. We like very much oscillating system where we have a set of the which we can with a proper description of interaction within complex system can control its dynamic behavior, and we are happy that we have results, and of course we publish these results in scientific journals, I in majority want to touch today the first object, but I will talk a bit also about the second, and usually I give a lecture about Lysigan creams as a new strategy for the material science, but let's talk about this ion concentration gradient system. What we usually try to do with our chemistry should be robust and sustainable. Of course, some materials is there, nanostructuring of the surface, to show some interesting behavior like swelling behavior, ion transport, storage, etc. Of course, we understand them on a molecular level, how it works. But after that, we have some brilliant patterns, like, for example, patterns made with cellulose to control its ionic channels. And in general, if we understand how the system is working, we find a number of applications, and we are happy that this application also can be given to artists, and they after that this robust system can make also look artistically, scientifically, because scientifically they mean a lot, because for example, one electrochemical platform, which is important nowadays, can be used to detect antibody antigen. They are complex. Of course, we are saying that it should be complex approach. We publish a paper, we collaborate with a lot of scientists, but we understand on a molecular level what is happening. We control some CVs, we build and use model like methods to analyze the data, to make some interesting things. But why I talk about the system of reaction diffusion, which I discussed as the first one, because if on a broader sense, if we are not limited to the scientific approach, we can start to look to the connection. And for example, dendritic system from the broader perspective can be found in various interesting areas. From the in a plane, unfortunately we are not using plane nowadays a lot, but in a longer trip I make some photos, and the mountains need some dendritic patterns. The tree build these dendrites, connection in the brain, dendrite slime mold, which Laura likes so much, making this, and bacteria colonies, if you feed them not properly, giving you some interesting pattern. And was famous scientists who also did a lot of exhibition concerning this reaction diffusion approach. But of course, the main in science to build this set of equation which allow us to predict the system. And then to build this equation to prove how it works, there are various model system to see these dendrites, and this model system we can give to scientists because they are working reproducibly well. If after that they try to make some exhibition of these complex patterns and how it works and how it grows, control the dynamic, and one of the system we, for example, able to grow, to control this light, to look for its beauty. It's totally chemical, and we try to understand what is the background of the formation of this dendrite for functional materials. But we're also happy when such a robust system which everyone can reproduce, and they are not toxic, they are friendly, but they have scientific meaning, can be used with artists to their exhibition. And we are nicely giving them, of course, as I mentioned, scientifically we are building models. We have a proper understanding how this model can predict our formation of this structure. But then we can start to think about some study where we control of the propagation of such dendritic network in a community, start this reaction for many times, control with other chemistry. But in the long run, I think all directions are interesting concerning the science, but I think this dynamic networks with oscillating reaction with a possibility of amplification, and since we have this control of the time, is interesting for people who are doing presentation of such system for scientifically meaning project, and we are always happy to help and to supervise and to give robust and sustainable system in their hands. And I thank you for your kind attention, and we invite people, of course, to read our scientific paper. We try to do them as clear as possible, but after that there are some systems which I think can reproduce people who are not chemists, and they can be used in some installation. And from my personal point of view, it should be, of course, important to find this like deep meaning in what the message we are sending to from our scientific perspective. If we in if we are taking part in such a projects. And my message that now is a time of dynamics, smart materials, and we can change in general the concept of material science to think about self-organization, self-growing materials, and we are happy to discuss this, of course, meaning.
Thank you, Ekaterina. And I think we already touched very important topics. For example, like the pipeline of the scientific research, and also what is important for scientists when they are collaborating with artists or in art and science projects. But hopefully we will go to it a little bit later. Now we have the voice of an artist and scientist too that has been working in different labs. And I would like to hear now from Lindsay your experience, your artworks, and so the public can also understand what is a science artist doing.
Yeah, for sure. Let me pull up my, yeah, so this is one of my most recent projects. I think I'm just going to go through my projects from most recent to least recent. But this is a work that I am still doing at Humboldt University of Berlin in the biophysics group of Hegemann. And so really what I'm looking at here is that there are these algae species called Chlamydomonas reinari, and they are light responsive. So they have these channel rhodopsin that open and close based on in response to light. And so when I first came to this lab, I was really interested in this concept of controlling these Chlamydomonas through light and their behaviors, because they have specific behaviors that react into the opening and closing of these channels. And one of the things that really kind of interested me was the notion of kind of bioengineering these pathways and what that means, because this definitely has a lot of implications for future technologies, as well as the field of optogenetics, which is kind of looking to sort of take these mechanisms and put them into the brain to basically control the function of different individual neurons is the long-term kind of expected goal. And thinking about controlling living systems is quite interesting, especially when we take into consideration that the living systems that we're studying this on are not human, and we're controlling their behaviors and genetically engineering them to manipulate these systems and pathways. One of the ways I thought that would be an interesting thing to artistically explore this was through this basically dance battles. And there's this arcade game that was quite popular in the US as well as in Japan in the 90s/early 2000s called Dance Dance Revolution. And so what I've done is I've taken different strains of genetically modified algae as well as the wild type algae, and I've created a song for them and choreography for them to dance to, and they basically compete against each other to see who can follow my choreography the best based on their different mutations. And so some algae they've been mutated so they can't really respond to the light at all, so they don't dance. And then some have a very delayed reaction, whereas some can follow it. But it really puts into context the things that are happening in the lab and with the genetic modifications on these algae species in a way that I think a lot of people don't understand, because when we think of genetic engineering, we think of a lot of control metaphors come to mind, and very like the way that we can control other organisms comes to mind. And what I found out through creating these dance battles is how uncontrollable the algae actually are, and how much genetic engineering really doesn't create that control within a living species. And it's quite interesting then to show this to people, especially audiences, to really challenge their perspective of what they think genetic engineering is, as well as what they think about how even algae is, because most people think algae as the stationary pond scum type of thing. So it's been really interesting with this project to see both perspectives on that, as well as I'm really excited to see where this project is going, because it's still in the process of development. now I can't change the, this is complicated. 1 second. Okay, did it slide change? Perfect. Okay, and so this is kind of the mechanism in which I was really designing this piece to go. These algae are flagellated and they're brought into a glass cuvette in which a light is turned on and off, coordinated into the dance choreography. And so I program it into the computer, and then we'll run these over and over and over again for the algae to dance to. And over time, the algae become less responsive to the choreography as well, because the light stimulation becomes too much for their photo receptive system, which is also interesting, because then you get this narrative about the organism and the material's own agency against my agency as the artist and as the bioengineer. And so going back to some of my earlier projects, this was a project that I did at Symbiotica in the Department of Human Sciences and in the Spinal Cord Repair Lab. This is a work that I was really exploring a lot during my thesis, the notions of materiality and aesthetics of stem cells. Oftentimes stem cells are given this aesthetic promising potential to them, and they're seen as a material that can just become anything, and that's often swayed to things like organs and stem cell therapies and all these sorts of wonderful promising things for our future, which is great. But the material reality of stem cells is actually quite different. Stem cells can also become cancer cells, and quite frequently it's not known what a culture of stem cells will become, and a lot of the characteristics that you select for within a culture of stem cells are the same characteristics that cancer cells have, which is very interesting. And it's quite interesting for me to think of the cultural context of why this is happening in science and why this promising potential is the one that's advertised to the general public versus the material reality. And so while working on this, as well as another project, I was really interested in exploring these material qualities of stem cells. And so the hanging drop became a tissue culture performance experiment that really aimed to question the ways that these cellular bodies have their own micro agencies of creating shapes and forms and all sorts of things. And so I would suspend these droplets of nutrient medium, which is a liquid kind of food substance for the cells, and the cells inside of it over time would create these different sculptural structures. And they would last for about 72 hours in total, and they would reach this peak sculpture making, which is when these photos were taken. And then right after that peak, the cells would begin to die because they would starve to death in these droplets, and the sculptures would start to dissolve. And when I then ended up exhibiting this work, that became an even more interesting conversation point for me and the general public, because oftentimes my authority as an artist to kill my art was being called into question. Who gave me the right? Why, as an artist, I get to kill these cells, when in reality, these cells were actually marked off as waste and were going to be thrown away anyway, the ones that I were using in my project, as well as scientists kill cells all the time in the lab for the simple reason that the culture in their flask has too many cells in it and they need to dilute it. So it became quite interesting that once I brought this into the gallery, this sanctity of life was really called into question, versus when in the laboratory, it's oftentimes not so much. And this is a close-up of one of the sculptures inside this droplet. And all of this work really kind of paramounted into this film I ended up making called Return of the Teratoma. And a teratoma is a type of tumor that grows teeth and hair. It's made of germline cells. Typically, it happens, the creepy ones that have teeth and hair typically happen in female reproductive systems, because when they're in a male reproductive system like the testicles, it becomes a malignant testicular cancer, which oftentimes kills the patient involved. But in female reproductive systems, they'll grow up to 300 teeth, I think they found in one of them. It's quite horrendous, their appearance and shape. And so I was kind of looking at how the figure of the teratoma has really returned in the laboratory, how we're creating all of these laboratory stem cell entities that are quite uncanny, much like a teratoma, and quite monstrous. And so this teratoma in the movie poster is a teratoma that I grew. It is over 180 days old. It's made from a retinal organoid, which was taken from embryonic stem cells, and it was grown in the lab to differentiate primarily into the retina tissue of the eye. So it has 2 little black dots for retinas, as well as I made teeth to attach to the organoid, and so it could have the teeth and hair element of a teratoma. But because the way that you differentiate stem cells in the lab, there's also a lot of unknown components that ended up being part of this organoid. So these bit fluffy bits at the end is hypothesized to be lung tissue, but it's not actually known what it is, which is quite interesting. And so in this horror movie, I showed the process of how I made these creatures. So they were taken from a culture of embryonic stem cell organoids, and the one I chose ended up having these wonderful distinguishing features that I think are quite cute now. I think it's quite a cute monster. And these teeth were made from cuttle bone that was centrifuged in laminin and then grown with mesenchymal stem cells to be reattached to the original organoid structure. And so in the horror film, the organoid ends up wandering through different parts of New York City, haunting actually my friend and collaborator on this work, Marc de Pasquale. And it really brings the context of this figure displaced from the human body. And during exhibition, people kept asking me, like, did I make that in After Effects? Did I digitally alter it? And I was like, no, that's actually the exact, it's a living thing. Those images come from when it was alive, and it's just been blown up really big digitally and placed in these very human contexts. Like nothing has been CGI'd in that regard. And it called into question the reality of what happens in science, as well as the reality of kind of these figures that are emerging from the lab themselves. I also had them, let me see if I can find it, in the gallery space as well, in little jars for people to kind of like investigate to see if they were real or not. And even then people were sure that I was making everything up and those were all staged props when they weren't. So it was very interesting to see people's reaction to what really emerges from the laboratory versus what they think should be coming from the lab with stem cell technology and stem cell biology. And as a side branch of this project, I really was exploring a lot the concepts of 3D bioprinting different parts of the body in relation to teeth, and looking at the different ways that teeth could be 3D printed and all the problems that come within printing these complex types of 3D structures. In the end, this project was too undercooked to be successful due to the fact that the cells to be pushed through this 3D printer with enough structural rigidity had to have this really cytotoxic material into the gel mixture that they were being printed in. Unfortunately, otherwise they would have collapsed. But it was definitely a very interesting experiment, especially to work with scientists, to push them to take this was a technology that they published a paper on, to really push them and think about creating specific structures with specific purposes and having specific cultural kind of entanglements around that. And this is an earlier work from my undergrad where I was working at with Dr. Joy Reidenberg at Mount Sinai Icahn School of Medical School. And Dr. Reidenberg is an expert in whale cranial systems. And so I was really interested in this kind of surrealist, postmodern theorist Bataille and his informe, which is the formlessness. And so he makes this argument that we must crush all forms of kind of taxonomy in it into nothingness in order to truly bring some more truth to the world. And these curators, Rosalind Krauss and Ivy Lanz-Boyes, came after that and tried to re-articulate how postmodern art is talked about and curated based on this guide. And they came up with 4 principles called base materialism, pulse, entropy, and horizontality. And they were reorganizing postmodern art through these 4 principles to have it express a different kind of narrative and restructure it away from form. And I thought this was really interesting to explore in relation to the heads of cetacean animals. And cetaceans are whales, dolphins, and porpoises. Because taxonomically, whales, dolphins, and porpoises anatomically don't make any sense. It's very hard to understand why they are able to exist in the first place. And so looking through these 4 different principles, I ended up creating these animations, as well as a dissertation that explained how we could restructure knowledge away from this evolutionary taxonomy that would still make sense in science and still have scientific value to it. These are close-ups of the different animations. And the last project that I have here is kind of a crime exploration. There was actually no crime that took place, but Mickey Meow was a beloved pet cat of Dr. Joy Reidenberg, who passed away at the ripe age of 16 human years. And I artistically ended up exploring the process of pathology and bioethics into solving this mysterious death behind Mickey Meow. So from cause of death to all the details behind it, to this, there was one point we weren't sure if the cat had magically cured himself of his diabetes or what had happened. And so all these different things were explored, as well as like, what are the ethics in presenting this information to the family of deceased, and how does death get explored when it's an animal versus a human, also when it's a pet versus just any other animal in the lab, which was quite interesting to do, as well with Dr. Virginia Gillespie at the Animal Pathology Department at Mount Sinai. And that is a brief overview of my work.
Thank you, Lindsay. It's awesome. What I can see for both presentation is just the different way to explore life. I think scientists and artists, that's what we are doing, just exploring what means to be now in this conformation and what we can do with it. And there are different approaches, but in art and science, somehow they are interconnected. I think art and science itself is trying to erase kind of the boundaries that humans have made into the disciplines. But I decided to make a list of questions that are the questions that I mostly also receive from general public, and sometimes they are the most difficult to answer. So I would like to hear, for example, in your opinion, for both of you, what is the main goal of art and science collaborations?
I think the major is, of course, to discuss with the community the importance of both, and to give this freedom of interpretation, because science is quite strict, because science should be the same results in many groups, and this is what we are about, to have a reproducibility, to have a reliable system, and simultaneously this reliable system can be thinking on another content and context, and we are happy to give it to the community. As a scientist, what I see, that of course I should follow first of all the safety rules, because you're also discussing some biological object, some in our case, chemical species, and I care that what we are giving to artists should be also friendly. And from my point of view, I expect that it's not horror movie or something. I expect that it will allow me, and honestly, this is very good. It's given really good results when we work with designers. That of course, to attract attention with something which people are afraid, easier with than something which they not understand. And here, I think it's interesting to really find this approach, not to be afraid of chemistry because it's friendly or biology, but since you understand there is a scientific meaning, and then this is for you the same object as various pigments. You are making a picture, and now your picture is making with some instruments. I am happy to share these instruments with the community. As you mentioned, you are using 3D printer, you're using this data, and the science now has a lot of robust and sustainable apparatus, and since we understand how to do it reproducibly, you're also thinking, oh, can we use it as a material for our art? And here I see for artists, us scientists as a supportive people, and if we're involved in this project, I of course care either it should be horror movie or something a bit enthusiastic, or probably horror movie, but with a better understanding that if you control this horror, it's not such much horror, because if you in a lab, what you care about safety, of course. And okay, the system which artists who are going to our lab, we are giving them instrument which have scientific meaning, which we can explain, which we think can allow us to communicate with biological objects. And after that, if they do their own understanding how to connect it with a social feelings, I think it's really important, because I think art is about feelings and science about the truth, which is like true for everyone.
So we could say that one of the interests from the scientific part is this communication from the work, from the safetiness, from the educational part. That's one of the interests from the scientific community working in art and science. But then we also have like this other part that is the artist with the ideas and philosophies. Lindsay, can you maybe tell something about the goal for your art and science projects?
Yeah, for sure. I think the goal for my art and science projects kind of it teeters between both the cultural sector and the scientific center, because on one hand, I do want to communicate with the general public concepts in science, because I think the way that science is rapidly changing the landscape of our future is really important to discuss, and to kind of imagine that future and bring that future into some sort of material reality, either through object making or through visuals or through an installation or some sensory experience. I think that is very important for the public, to bring them in that dialogue and to like make those futures into a sort of reality for them to explore. But on the other hand, for me, and the thing that I think I find the most interesting about being an artist and working in labs is kind of the questions that I end up bringing to scientists about their work and about the way that they work. I think that oftentimes science sees itself as an objective discipline that has wonderful rules and that makes it work, but at the same time, I don't think that it's always present in a scientist's mind when they do every single experiment, the cultural impact and ethical implications that it may have. And so for me as an artist, being in a lab and being able to pose these questions in this sort of interactive dialogue is really interesting and cool to have with the scientists, to be to bring in sort of more these ethical considerations, both historical and future ones as well. And yeah, and I think I learn a lot from scientists as well in that regard. I think they teach me a lot, which is really cool. Because I mean, I have my expertise from academics in science, but from zoology, and then I have my master's, my academic expertise from stem cell biology. So every single lab that I enter now afterwards, it's kind of becoming a new expert in something else. And so I end up learning so much. And so for really me, the main goal of the science art projects that I do is to both not only communicate and imagine the potential of these relationships and these futures, but also reflect on like how science as a cultural body has really impacted society and people, and tell those stories both to scientists and to the general public and to other artists as well. Yeah.
Thank you. I think you already also mentioned a benefit that the presence of an artist make in the lab, making questions from a different perspective from the same, from the work that they have been sometimes working for several years, and then comes an artist and make the proposal of sentiments or emotions, and that can also change their point of view. Ekaterina, I would like to ask you to, what other benefits do you think scientists can get from working with artists?
I think it's of course in general for older scientists, it's just another perspective, but for younger scientists who just started, I think it's really another way of thinking. It's looking for the object from various perspectives. And as I mentioned, science is not just my work, it's also my hobby. And if I think, as a older person, I can make my own definition and further discuss with you. Of course, when I talk with my colleagues about my science, I'm more talking about facts and the problem, the approach, results, and that I try to present them. But simultaneously, my science also is beautiful for me. I like feel sympathy to I like my science also personally, because I studied this system 24 hours, 7 days per week. And of course, it starts to be a part of, not just life, which is life professional. I want to show this beauty and to show that this dynamic demonstrates something. And here, of course, artists help to go deeper, not just to show that it's beautiful, but to understand what is a basic key, why this beauty, what philosophy is underneath, who else are thinking that this object, and okay, some chaos, like self-assembly or making from chaos some ordered structure, how it happens, and that this I always surprised to find out that something which we try to describe as a formula can be nicely connected to something which people describe in a philosophical manner, and it's a deep philosophy underneath, and people spend also a lot of efforts to find out how this chaos, what is this chaos, and this understanding helps also to educate the younger generation, to make them, because I think it's really helpful in the context of science education through science. It's needed for the younger generation.
Okay, so then we are talking about changing in the research and also education, but there is also challenge. So then an artist can, that a scientist give time to create some artworks, or the facilities or the money to not make just scientific research, but also artistic research. Lindsay, have you have some of this challenge to open the space for your work in the laboratories?
Yeah, I would say money is usually the biggest challenge, but that's often the biggest challenge in art ever, is to find the money. Also the biggest challenge in science too. Surprisingly, science and art have a lot more in common, I think, than they like to admit in those regards. But as for me personally, because I'm very lucky where I do have this background in science, like heavily in science, and I've worked for so many scientific institutions at this point, and I've been trained that I usually don't have a problem being able to open doors into scientific laboratories because I'm as skilled as a technician could be in many things, which is quite nice in that regard. I think a lot of other artists in the field don't have that same sort of privilege to be able to do that. But finding money is always difficult. And I think luckily now there are more transdisciplinary focus, especially here in Germany. I think there's becoming more and more transdisciplinary focus, which is really nice to see, where both the government and the European Union really want to focus on having art and science talk to each other a lot for science communication, because that is a main benefit of having these exhibitions and things like that, is getting the public to start having interest in these sort of main scientific concepts, whether they're questioning them or debating them or having some sort of inner conflict or supporting them, whatever it is. I think having that interest being spurred in the public is really important, which is really nice to see. Yeah, I think that just the money part's the biggest hurdle to get over. And once you get over it, then most scientists are always welcome to have someone getting their hands dirty, as long as they're being respectful, of course, and not contaminating the lab, which comes with skill and training. So.
Okay, and thank you. I think that one of the benchmarks in art and science research is, for example, what you show, that the scientists publish a paper with the technology that you were using to make your artwork. How does that work? Is it easy to publish art and science collaborations? Where to find these magazines? Can you tell us some?
Yeah, so with that one in particular, the paper was published and then I used it in the artwork, but sometimes it's quite easy. There's a couple of journals. Leonardo is one in particular that is very much a transdisciplinary art and science publication, where you can do anything from protocols to artworks and things like that. But also, I think the Journal of Artistic Research publishes a lot of art papers that then you can add science things in there as well as the Journal of Video Experiments now is quite interesting in that regard too, because you could easily publish a lot of artistic protocols that have been done in science labs or scientific protocols with other art collaboration, as long as it's in a video format to the journal. I could definitely see that being a place. I think the landscape of publishing is changing a lot though too, in terms of transdisciplinary research, because peer review becomes then quite complicated, because you need the right peer reviewers to review it, and they need to then have transdisciplinary expertise, which there's only so many transdisciplinary experts in the world, or you have to have this like integrated jury. So what I've seen as well, working with Bohr Collective, which is a Dutch bioart collective, is that there's been a lot of like more online publications in not necessarily journals, but on platforms like Bohr and like these sort of blogging and DIY bio movement spaces, which has been very interesting to see happen as well. But I would say on the tail end of that, the exhibition is the equivalent to a journal publication for an artist. So there's also that as well, scientists having their own exhibitions and things like that.
Mm-hmm. Ekaterina, do you feel excitement about the exhibitions, like seeing the artworks there? Do you think scientists will also share this excitement?
I think it's the most interesting thing when you see, because my experience was we also publish a scientific paper first, and after that we are ready to give this as instrument and an inspiration. And as I mentioned, you also mentioned this that you have experience to be in the lab. It's important that okay, if someone come into the lab, you of course much easier having in the lab someone with previous expertise who at least know how to use pipette, because okay, safety is a big issue in the lab. And of course, if someone come in not for just to see us, but to work with us, they expect that they know the rules. If it's some system which I want to give for exhibition, as a scientist, I also take care it's safe, first of all, it's not toxic, it should be, okay, I still could not kill this, okay, me, of course, it should be safe. And after that, it should have some contact. After that, it's nice to see really like my inspiration from the artists is how much effort they are putting in really making final exhibition, because this should be not just an idea. You should create something, film or sculpture or some object which people interact with. And in my particular case, I see immediately that materials which we develop can go to the market. It's from the point of view of money, it's much easier, as you mentioned. But here, the people like so inspired with something that they are ready to spend a lot of time, and they still don't know either it's going to be useful, how other people feel about this. And I am happy that always to see how much attention and how people happy to interact with it. At least it was during this open space communication in ITMO. And I'm happy we have now this place for installations and communication.
Yeah, I also think that it's very important to have these spaces. And so I don't want to take more of your time. I just want to make the last question. If you can summarize in an advice for the artists and scientists, how to make a successful collaboration. For example, if an artist comes to you, Ekaterina, or when Lindsay, you are asking for collaborations in the labs, what you can suggest for them?
Yeah, my first suggestion would be to pick your science medium carefully. Don't just approach any lab, and when you do approach them, come with a deep background, like research done already, because I find that when you the main thing about having successful art and science collaborations is being able to ask very, very interesting and profound questions to the scientist. And so unless you already have some sort of background knowledge you've built up through reading journal articles and publications and things like that, you really need all that background knowledge to be able to ask these questions to make your artwork. I wouldn't say that you have to be scientifically trained as an artist to enter the laboratory space. There are plenty of wonderful and fantastic artists who are no scientific background who have entered the laboratory, made fantastic works, and have been very successful. But it's more just coming with this respect for the space that you're entering and respect for the scientists, especially when it comes to things like safety. I think that's a big important thing, which if a lot of artists who've gone to art school know a lot about safety, especially if you work with big power tools and construction sort of equipment of huge installations, like safety is a priority. You can't just enter a space and act unsafe. But coming with a lot of research being done and picking the area of science you want to go in very carefully, as well as just like having a positive attitude, really, because you're going to get a lot of doors shut on you at first. It takes, but once you find that one scientist who has like, who's a little bit interested, who goes, oh, you're weird and different, but I kind of like what the questions that you're asking, let me keep you around for a bit, then I think you're all set. But it takes time, and you have to do a lot of work on your own to like get yourself there. You can't just expect that a scientist will let you in their lab because you're an artist.
That reminds me about the science speeding date that the marathon in art and science were making this weekend. So they were trying to match artists and scientists to find your perfect scientist to work. And I think, yeah, it's really important. I don't know if Ekaterina, would you like to share something about the suggestions to work with art and science collaborations?
I think it's really important to be open-minded, to listen each other, and these interesting questions from both directions should be not just asked, but also we should hear them and spend some time to think on them. And this is what I'm teaching also my students, because if scientific supervisor says that it's interesting, it's of course should be some collaboration between they should be interested in this idea of interesting questions, because in a major thing it should become the big part of their life to think about their scientific object all the time, 24 hours, 7 days per week. Just in this case, they are successful in science. And I think in this case, they are happy to share their knowledge with artists. And we are very thankful to artists for their questions, of course. Ask more. And it's nice how you imagine it, that you are working on them, you are thinking creative, not to be like to make people afraid of science, but to explain them the deep meaning and to give this as instrument to them how to do everything. Thank you for invitation, Laura.
Thank you both for attending. And I just realized something very interesting, that both of you are in the labs, right?
Yes, today I am.
So, although we are sharing a little bit of the difference and so on, there is a lot of similarities. And I hope that during this talk, our public can understand a little bit more of what it means to be an artist working with science, and why it's important also for scientists working with artists. Thank you. Thank you for your time. And that will be all. Thank you again for this time.
Bye.
Bye bye.